Lovely. Well, thank you for coming along, Sandra. Lovely to have you here. A bit of introduction about Sandra. Sorry, I can still hear some background noise. Could everyone make sure they're on mute, please? Thank you. Hang on a second. Someone's not. Let me just. There we go. Okay, lovely. Thank you. Okay, so kicking off about Sandra. So Sandra is currently a bioinformatician at the University of Queensland at the Diamantina Institute, and her focus is on esophageal cancer. In 2015, Sandra had the opportunity to return to Australia to complete her PhD in esophageal cancer genetics. And we're delighted to say that we're funding her this year. This is the first time we have funded Sandra, so welcome to the family. And it's great to have you here as part of the Cancer Australia Priority-Driven Collaboration Cancer Research Scheme. So Sandra, before we talk about your research, do you want to just explain to everyone here a bit about you and how you, in fact, got involved in cancer research right at the beginning?
Sure, thank you for having me in the first place. So I'm Sandra and I'm originally from Germany. And so I've done my bachelor and master's degree in bioinformatics and genome research. And during my studies, I was always like, bioinformatics is a very broad field. So we got to test out a lot of different areas and fields. And I never really found anything that was really fascinating me or that was really tickling my fancy sort of. Like it's always like, yeah, it's okay. I can see myself doing this, but it's not really what I'm after. And then so for my master's project, I had the opportunity to actually come to Australia and Brisbane and do some medical research. And I was really fascinated about it because it's challenging, it's frustrating on bad days, but it's really rewarding when you find something and you can sort of like, you can see how that might affect patients in the clinic. And it really fascinated me. And I was like, yeah, this is what I want to do. So I don't really have like a personal story to tell of like, oh, I have personal experiences with this cancer type or something. It's more my curiosity and my feeling that I want a challenge in life. I want to do something that also impacts people in a positive way. And that's how I pretty much got stuck where I am at the moment, and I really enjoy doing it.
Brilliant. Now we at Cure Cancer, we are the only organisation that funds bioinformaticians in the ECR stage. It's a long word. Could you explain to everyone here what is a bioinformatician?
Yeah, that's actually quite a tricky question. And one that I get asked a lot, and I'm always like, yeah, so bioinformatics can be a lot of different things. So there's, you can pretty much split it into two big groups. There are bioinformaticians who develop tools, so they can then be used in a medical or in just a general research environment to analyze data. So they're on like the tool development things. They implement pipelines to analyze all these big data sets that we're now having. And then there's a second side of it that are actually people that are using those tools and that are analyzing those big data sets. And that's more where I see myself. So I'm using existing tools and programs to then analyze big data sets. So it's pretty much a data analysis type of thing, a little bit of statistics, but we're not statisticians because that's also something that often gets mixed up like, oh yeah, you're a bioinformatician, you have to know statistics. Well, I do, but not to the extent of an actual statistician. Yeah.
Great. And so what would your background be? Could anyone start bioinformatics, or do you have to have a numbers or numerical background to get into this career?
Look, it helps if you like numbers, because that's pretty much what you would be looking at the whole time. Like you'd be sitting in front of a computer and you're just looking at numbers and letters and stuff. But you don't actually need like a formal education in this area because so we currently have a PhD student and he's a clinician. He's using R on a daily basis now, and he's analyzing his data sets fully by himself. So with a little bit of training from experienced people around you, it's definitely possible to get into it, even if you don't have the actual education in it.
So does that mean then that you don't spend much or any time in a lab and it's all in front of a computer? Is that right?
Yeah, that is correct.
Okay,
Which was really handy during COVID times because it was very easy to transition to working from home because all I need is a working internet connection and a computer.
Yes, yeah, and you know, we funded a lot of researchers, particularly down in Melbourne last year, and some of them were locked out of their labs for 7 months or 8 months. So yeah, very handy indeed.
Yeah.
Lovely. Now let's just talk a bit about, before we talk about your actual research, but esophageal cancer, which is your area of speciality. Why did you decide to get into this type of cancer research?
I mean, to begin with, it was more a coincidence because that was the projects that were available for my master's research. And then I just pretty much stuck to it. And I was like, this is really interesting. And because it's such a understudied cancer still, even though it has a very high mortality rate, there is still so much we have to investigate and so much we have to analyze and learn from it, that it's still a big field that you can spend hours and hours and years of research on.
So let's just talk about the locale of it first of all. So when you say esophageal cancer, are you talking about all of the neck as well, or a bit more than the neck?
So there are two main types of esophageal cancer. One is esophageal squamous cell carcinoma, which can be located anywhere in the throat because that's mainly occurs because of smoking and alcohol consumption. The one that we're focused on is esophageal adenocarcinoma, which is located right at the bottom of your esophagus. So it's right at the end, like at the connection between your esophagus and your stomach. And so a lot of it happens because the gastric reflux and the acids from your stomach go up, damage the cells there, and that's how the tumor often develops. So it's more at the bottom of it. So
at the bottom, yeah, that's really interesting because that was going to be my next question. So it's purely acid reflux, or can you get this type of OAC through other means as well?
You can get it through other means as well, but most of it is associated with reflux disease. Okay. So that's a really big risk factor for it.
Yeah, okay. And what would these symptoms be? How would they present themselves in a patient that may have this OAC cancer?
That's another tricky one because OAC usually gets developed very late stage. So most patients are already at stage 3 or stage 4. Their symptoms aren't very severe at the beginning. It's more towards the end, or if they're getting checked out for their reflux of like, oh, look, I'm really struggling with this. Can we have a look? And then it actually occurs that they already have a cancer developed. So I think the distinguishing between the cancer symptoms and reflux symptoms is not very big. So that's why it gets discovered very late stage usually.
Well, that's sad, isn't it? And you talk about a high mortality rate. What are the numbers around OAC?
I think the 5-year survival rate is up to 10 to 15% now. So that's very low in comparison to other cancer types, unfortunately.
Yes, absolutely. So why, I mean, obviously, if the mortality rate is so low, or survival rate so low, why is this research underfunded?
The case numbers are not as big as breast cancer, melanoma, or the lung cancer. So they are more common than OAC. Yeah. And the other problem is also, I mean, esophageal cancer might be common, but most cases are the squamous cell carcinomas, not the OACs. Yeah. So you're dribbling down and it gets the sample sizes get smaller and smaller, the case numbers get smaller and smaller, that not a lot of groups are actually focusing on this cancer type.
Okay, all right, well, thank you for that. So let's really talk about what you're doing and the analysis, the bioinformatics you're doing to help with this cat. So do you want to kick off and explain what your current project is that you're working on?
Sure. So one of the main problems with OAC is that we don't have a perfect treatment for patients at the moment. Because for a lot of cancer types, we have targeted therapies, we have mutations that we can target, we have a lot of drug targets that can be used. But in OAC, we don't have that. So the standard of care is still basic chemotherapy with or without radiation. And then after that, usually surgery to remove the tumor. But it's a long time to recover from that because you're taking a piece of your esophagus out and you're moving the stomach up. So to recover from that surgery, it takes up to a year because it's a big impact. And before surgery, only 40 to 50% of patients actually benefit from chemotherapy. But everyone who goes through it obviously has the potential side effects of it. Yeah. So that whole thing is not it's not ideal. So if we would know at the beginning when a patient presents with a tumor, if they would actually benefit from the treatment or not, we could then say, look, your genetics is not good for this treatment. We send you straight to surgery because we can't do anything for you at the moment otherwise. Or we know this patient would respond very well to chemotherapy. Yes, you might develop some side effects, but it will definitely benefit you. So that's sort of like the big problem that we're having at the moment, that the standard of care is effective in some, but not all patients. And how do we find the patients that will benefit from it versus the ones that won't benefit from it? So that's the big thing that we're trying to target with our genetics approach.
Yeah. So how do you do that? How do you start to look at trends to see that I, as a patient, might benefit from chemo versus you, as a patient, need to go and have surgery?
So what we do if a patient comes in, we ask, like, they get an endoscopy done and samples will be taken during endoscopy. And we use those samples, we send them to sequencing. And then, I mean, for the patients that we currently have, they won't really benefit from it. But in the end, we have a patient cohort with patients that respond and patients that don't respond. And then we're going to use the data and compare what is different between those two patient groups, that then if a new patient comes in, based on which group their markers fit into, we'll know what to expect for them.
Great. And when you say markers, what do you mean? Could you explain a bit more around that?
So that could be pretty much anything. So that could be a specific mutation in a gene. That could be a specific expression profile of genes. Whatever we sort of feel like, yes, we can reproduce this. This is a valid finding. Ideally, it's a set of mutations or a specific expression profile of genes that we would use.
Okay, and then is that just the two options at the moment then? So once you have done this study and you identify trends, is it chemo and or surgery, or are you hopeful that this will go to lead into developing different drugs that are going to work in certain patient groups?
That ideally, if we find a drug target, for example, in the patients that currently don't respond well to chemotherapy, that would be ideal. If we could say, look, you won't be responding to chemotherapy, but this drug actually targets your genetic profile, you're going to benefit from this. And there are studies at the moment, or clinical trials on immunotherapy drugs as well, which are promising in some cases. Again, unfortunately, not all cases. So that would be a follow-up sort of once immunotherapy is a little bit more established as a treatment in OAC, to then do the same thing. Can we find differences in the patients that respond versus the ones that don't respond from the treatment? Okay,
great. Just a reminder for people, if they want to ask questions, I've had a couple, but just pop them in the chat and I'll feed them into the conversation. So we were talking to Dr. Jan Janathini last week about prostate cancer and aggressive prostate cancer, and she was saying that there are different actually races that have a more predominant likelihood of having prostate cancer, which was of interest to all of us. From your research so far, are you finding in the genetics, is it hereditary or is it race related? What are the breakups of what you've seen so far?
It's definitely gender specific. So we see way more males than females presenting with OAC. So in my current cohort, I have 29 patients. I have 1 woman and 28 men. So it's usually middle-aged to old white males that present with OAC in the clinics.
Interesting. And maybe it's too early to tell, but do you have an inkling why that might be the case?
No idea.
No idea. Oh, good luck.
There are some thoughts that it might be related to diet, but that's, I don't really have evidence for that.
No, not yet. No, okay, understand, you're right at the beginning of this. And then in regards to genetics as well, has there been any predisposition to show that you can trace that along family lines as well, or is it purely sporadic at the moment?
I think it's more sporadic than as like a inherited thing. But that would, to really draw a conclusion there, we would need to do more like family studies and like really look into specific families where we had a case, and then go back in time, like, oh, do you have any other family members that also had it?
Yeah, understand. Okay, thank you. So you're talking about your specific cohort that you're looking at the moment. So just talk to us through your day-to-day role there. What do you do, and do you just analyze things all day, every day, or are there other aspects to your research that you do?
I'm mainly doing analysis pretty much all day. It's different types of analysis pretty much every day. Like, I mean, not every day, but every now and then. It's like you start with a big data set and it's pretty much just numbers. And then you run one analysis on it, and then one leads to another one, and it leads to another one. So you're sort of like consecutively finding things, and then you're basing your next analysis on that. But my daily work is pretty much just analysis.
Yeah. And do you, we ask this question of all our researchers that we fund, are you working with other groups within Australia or internationally as well on this research?
We have a collaboration with a different institute. Actually, she's on the call, Nick Waddell.
She's just asked a question as well.
What a good timing. So we do collaborate with other groups, but there are not a lot of OAC research groups in Australia. We had a clinical trial that was collaborative between multiple hospitals across Australia. So we got samples from all the hospitals from different sites, but it's more the sample collection rather than the analysis that we collaborate with.
Interesting, thank you. And a question from Nick. She was just saying heterogeneous, which means the genetics of a tumor can vary within a single tumor, which makes very specific targeted therapy difficult. So what work are you doing on heterogeneity, and what results are you seeing so far on that?
So that's one big part of this project is, so we have the tumor, and then at endoscopy, they usually take apart at the top, the middle, and the bottom. And in most cases, you send one of those pieces to sequencing and you have a look what is in there. But we found that OAC is very heterogeneous. So the top part looks very different to the middle part and to the bottom part. So what my project is trying to establish is how different are they actually? So we use multiple samples at the same time. So we're taking two pieces from endoscopy, send them to sequencing, and then have a look what is common between those pieces of tumor. And those are probably the changes that we want to target with a targeted therapy, because it would target the whole tumor and not just a little piece of it. And then also what are the differences between them, because what happens to one part of the tumor but not the other part? Like what drives these sort of like subclones to change all of a sudden from the rest of the tumor? So that's a big part of my research. And the other aspect is, once a patient receives chemotherapy, that also changes the genetics of the tumor, because chemotherapy changes, it changes the DNA in the tumor cells, and ideally it changes, or it actually destroys it enough so that the cells die and the tumor, the cells disappear. But quite often it introduces changes to the DNA, but it's not enough to actually kill the cell. So those changes over time because of the treatment also need to be taken into consideration when you treat the patient, because what you see now is not the same what you saw at the beginning of the treatment. So we've taken samples at surgery, which is after the patient had their chemotherapy. And again, we're sending them to sequencing and we're looking, okay, what are the changes now? Have we overlooked something in the pre-treatment samples that now came through because some cells died, and the other cells had more space and resources to grow? So are there specific cell populations that now are here in a way bigger proportion than at the beginning of the treatment? So
that's really interesting. I mean, I can't imagine your samples are that big that you take. So is it common to have different genetics within the tumor or the cell sample?
It really depends on the tumor type, like on the cancer type. I think OAC is one of, I mean, I don't have experience with many cancer types, but from what I've read in the literature, OAC is very heterogeneous, whereas other cancer types are less heterogeneous. Like it's more a homogeneous mass of cells, and they all look very similar. And that's probably also why targeted therapy works well in some cancer types, because you're targeting the whole population of cells.
Yeah.
And in OAC, it doesn't, because you might be targeting 20% of the tumor, but the other 80%, they can still grow. They can still do whatever they want.
Yeah, that's really interesting. I wasn't aware of that. Well, thanks, Nick, for that question as well. That's interesting. So I have a question here about the treatment then. So obviously the part of the esophagus that you're dealing with is, as you say, close to the stomach. Having repeated samples of chemotherapy on that can have, I imagine, some pretty devastating side effects to people as well.
Yeah.
So what are the most common things that you see from people that have had chemotherapy and it's not working from a side effects perspective?
That's a very good question, because I'm not a clinician. I struggle to answer that question.
We'll have to save it for a clinician.
I mean, I can speculate, but that is outside of what I'm actually dealing with, because I don't really see the patients in the clinic. So all I get is pretty much an Excel, a big Excel spreadsheet with numbers or letters, and I don't have the day-to-day patient impact or like the patient interaction to actually answer that question properly.
No problem. We'll take that for another person. Okay. So just carrying on about how the genes change as well. So if chemotherapy is changing at different rates at different times as well, I mean, that's a really big puzzle to solve. The amount of statistical analysis you have to do to find trends, etc., is probably part of your challenge.
Yes, I call it frustrating on bad days and challenging on good days, depending on how things are going.
Oh, I can imagine.
Yeah, it's a big puzzle, and there are so many little things you have to take into consideration, and yeah, it's challenging. It's a good challenge. It certainly keeps us on our toes, and wrapping our head around things is not always easy, and making sense out of things you see is yeah, it's a fun experience.
So on that line of challenges, I've got a question here about what are your obstacles you encounter on a day-to-day basis? And part of this question is, well, what is it about the internet, slow internet here, or poor access to data sets? What do you find the most challenging thing, apart from just trying to sequence genes?
Right. I think one of the challenges is our sample sizes usually, because, I mean, the first issue is that OSC is not the most common cancer type. But even if you have sort of like enough samples in your tissue bank, like just in the freezer somewhere, finding the fundings and the money to actually sequence them and to then have someone and analyze them, it's quite like, that's a very limiting factor. So we usually we do what we can with what we have, but I mean, a sample, like having 29 patients, yes, you might see a trend, and that might lead into, okay, we can try and get more fundings and we can find more samples and do more analysis. But it's very often, it's like a discovery thing, but you don't really have enough evidence to say this is definitely something. You would always need follow-up studies with a bigger cohort to then validate your findings. So that's definitely a challenging thing on a daily basis, because I often don't have the statistical power to actually say this is a real finding and not a false positive or just a trend that we can see.
And an expanded question on this then, where are your samples from? Are they Australians? Are they international? Are they from 30 years ago? Are they from yesterday? Talk about your sample set.
So it depends. I mean, they're all from Australia. They might be from different hospitals in Australia, depending on which project we have them from. We very often try and reuse samples that we've already sequenced. So we're pretty much like, if we get new samples, we're usually just adding them to the ones we already have to increase our sample size, because we might be able to sequence 10 new patients or 10 new samples. That alone doesn't get us anywhere. But if we combine them with the 20 we already sequenced earlier, that already gives us 30. So yeah, but if it's for a specific project like mine, where we have a very specific question of, okay, we want to compare pre-treatment versus post-treatment or during treatment, then we have to make sure that we follow up with the patient as well to get the post-treatment sample from the same patient that we already have sequencing data for. And those would usually be newer patients, so from the last couple of years, but we also have samples that have been sequenced more years ago. And because, I mean, the beauty is, because my supervisor is a clinician, he actually has, like, he sees the patients, he takes the samples, and we have established a tissue bank where we can now just go and be like, okay, I'm looking for this group of patients, like these markers, like clinical markers, and then we just pull out the samples and be like, yep, okay, these are going to sequencing now. So that's very handy to have.
Yeah, no, absolutely. And what numbers do you need to make your findings statistically valid? I mean, are they, obviously the cancer type is small and it's low in numbers. So what are we talking 10 or 20 or 5,000? What do you need?
More in the hundreds. I'd say a good sample size would be 100 to 150. That very often already gives you good results. Anything lower, I can see a trend. That's usually how we put it. And like, I can see something that looks interesting, but I would like to validate that in a bigger cohort. And sometimes you can actually do it because you don't need to fully sequence the tumor in a bigger sample size. If you know what you're looking for, it's easier to find it in a bigger cohort, because you can target your analysis to look for just this very specific thing. But if you don't really know what you're looking for, your sample size usually has to be big so that you can draw conclusions from it.
Yeah, no, I understand. One of the questions we ask our researchers is, the work that you're doing, how can that be used across other cancer types? But it sounds like yours is quite specific. So is there an opportunity to extrapolate that out, or is it quite specific in its research?
I mean, what we try to do is, if findings were made in a different cancer type, we always try and apply that to ours as well. Because OAC is so unique, we often find that it might work beautifully in 10 other cancer types. It doesn't work for us. Yeah. But at the same time, there might be other cancer types that are also understudied, or that also don't follow the nice ways. They might benefit from what we find in OAC to then be like, okay, it worked on a cancer that usually nothing works on. Maybe it works on ours as well. I think it's not guaranteed, but it's always worth a shot to double check if something that works in one cancer type also works in a different cancer type.
Yeah, no, absolutely. All right, well, let's just talk about funding. And whilst I'm doing that, if there are any other questions, please just drop them into the chat. So obviously first year you've been funded by us, which is great. Can you talk about how important that funding is as an ECR? What does that mean to you?
Very good question, and very great that my project got funded. That's a very big step for me, because I'm 1 year out of my PhD. I finished my PhD end of 2020, and having that sort of first career boost of I'm getting a grant, I'm able to show that I sort of can stand on my own feet. I'm able to attract funding. My research is valuable. That is a big asset for anyone, especially in those early career years, because as an early career researcher, we don't have many papers. We haven't been invited to many conferences to give keynote speaks. We can't compete with someone who's been in the field for 30 years. We don't have that experience. We don't have the track record. We don't have the leadership to show. So having those very specific early career categories of funding is a really big advantage for us to be able to find our spot in research and to continue what we all love doing. Yeah. And
we've talked to quite a few researchers now about the amount of times that they take applying for grants, and as the more senior you get, it doesn't go away. But can you talk about how that works out for you? Grant applications versus actually doing what you're paid to do, so to speak. How's that time frame work?
So because I'm very early, I haven't been that much involved in grant writing, but I certainly observe around me. So we have 2 senior postdocs in my lab, and I think since January they've been pretty much only been writing grants. So there is time to do a little bit of analysis here and there, write a little bit on a paper, but pretty much 80% of the time is just spent on trying to find grants that you can apply for, preparing the applications, trying to work out all the budget and all that. It takes up a lot of our time, unfortunately.
Yeah, it's so sad, isn't it? Because you've got the brilliance and the brains, and instead of doing what researchers are meant to be doing, they're writing applications. And we talked to Jana last week, who had actually moved out of cancer research from Brisbane down to Melbourne to mental health, because mental health gets a lot more funding than cancer research does.
I'm not that surprised, yeah.
Which is an ongoing challenge. And what about bioinformatics? What's the funding like in that sector as well?
Very difficult, because like you said, I think Cure Cancer is one of the only ones that specifically targets bioinformatics. We're often part of grants, but we're usually, or quite often, we're not the CIA on it, because bioinformaticians, we always need someone else to collect the samples for us, to actually generate the data, to then be there for us to be analyzed. So it's very rare for us to get our own funding as like an individual researcher. It's usually as part of a team. I mean, all the big projects need a bioinformatician to handle their massive amounts of data that they're generating, but it's also usually down the track. It doesn't start right at the beginning of the project, because you're still getting your ethics sorted, getting your patients recruited, collecting the samples, sequencing or whatever you're doing with them, until you actually get to the stage where the data has to be analyzed. So that's usually only down the track, and yeah, bioinformaticians quite often get overlooked a little bit in those areas as well.
Yeah, no, I understand. We're delighted to say we're funding 3 of you guys this year. So that's
and we're all very, very grateful for that.
Lovely. So we talked about a bit of the challenges of what you do and the puzzle.
Yeah.
But what's the most rewarding aspects of your role?
When you actually find something, and you find something and you're like, oh, does that make sense? And I mean, a lot of the analysis, it's like a data analysis. And then you always have to take a step back. Okay, so this gives me a significant p-value, but does that actually mean something? So finding then the biological meaning behind it and be like, hey, this actually makes sense, not only on paper, also in biology. That is a very rewarding feeling if you can solve some little puzzle pieces every now and then. Yeah.
I can imagine. And then how does that go back to the clinician? You know, you've seen something, you've done an analysis, you're statistically valid. Then what happens then with your findings?
I mean, we would publish it in papers, but from there we would then try and set up a clinical trial to see, okay, it's sound on paper, it makes total sense, we can reproduce it, it's validated in like a very controlled environment. Now we need to go and actually do a clinical trial and see, can we actually change something in the clinical setting for the patients? That would be the next step then.
Yeah, lovely. Thank you. And then just looking into the future, you know, what are your greatest hopes? Where do you want to be in the next 5 to 10 years, and what do you want to be seen from the research you're doing?
I mean, obviously, I would love to cure cancer, but I think being realistic, that won't be the case. But I would love to be able to unravel a little bit of the mystery of the heterogeneity of OAC, because it's a very dramatic cancer type. It's very low survival rates, and if we could just help 1 or 2 patients to get the treatment that actually benefits them, that would be a massive reward for me in the end. That would be really good.
Yeah, thank you. And just a final question before I summarize our chat. What more can we at Cure Cancer? You've got the whole team here and some of the board. What more can we do to help support you?
I mean, I think the work you're doing is already great. And obviously, more funding is always appreciated, and funding opportunities, especially for ECRs and bioinformaticians. Yeah, I think just continuing the good work, spreading the word, that is all very, very helpful to us.
Well, lovely. Wonderful to chat, to meet you in this little square box. So really just to summarize, we're saying roughly the 5-year survival rate of patients with OAC is 10 to 15%, which is really low. Obviously a bit of a rare cancer because the incidences are low. The type of esophageal cancer you're looking at, OAC, is the one lower in the esophagus, linking to the stomach there too. And you're saying that only around 40 to 50% of those that do the standard chemotherapy treatment actually find this to be beneficial as well. And obviously we know the side effects, so we'll save that for a clinician, the side effects of chemotherapy. And you know, your hope is to be able to look at trends in the data to find out specifically what types of treatments would be better for different types of people as well, which is great. And the most interesting piece to me was of talking about the heterogeneous state of it. The fact that such a small sample can have so many different variations is really interesting. So thank you for sharing that. It's been a pleasure to chat to you. Thank you to everyone else on this call. We've got a break next week because I believe it's Easter, but we will be back on the 20th of April, and we're talking to Dr. Shafiq Syed and his work on uterine cancer as well. So thank you all, and thank you, Sandra. Lovely to talk, and have a good afternoon.
Thank you for having me.
Thank you.
Bye.
Pleasure. Bye-bye.